Thanks, everybody, for joining me here. I'm glad that Mitch mentioned the fact that it's been relatively recent, because all the work I'm going to show you has really been generated over the past couple of months. The premise at work here, I hope this is, whoops. Well, I seem to have shut off the talk. There we are, okay. There we are, is that you should be able to mount a response against your own tumor, because if tumor is a disease of genes, and if there's a mutational process that we have been focusing on driver mutations, that same stochastic process also generates passenger mutations. And your immune system is uniquely able to recognize cells based on the genes or the mutated genes that they express. So basically, if we're looking at the lack of tolerance for a target antigen, you should have very profound tolerance to your self-antigens that will leave you a repertoire of T cells available to see the world of pathogens. If you look for tumor expression of antigens, well, the tumor does express self-antigens, but so does the normal tissue, so we put this in the lower left-hand side. If these mutated driver and passenger antigens are uniquely expressed in the tumor, then you should have a repertoire of more or less equal size to that available for neoantigens that could potentially recognize the tumor-specific neoantigens. So that's the premise that we're pursuing. If you look across a variety of tumors of different histotypes for the number of mutations per megabase, you see that some cancers have very high levels of mutation, melanoma being the most antigenic cancer, and the one cancer among these at the right-hand side of the graph that respond very well to checkpoint blockade that is commensurate with their high mutational burden. There are other cancers, perhaps head and neck cancer, the one that we're studying, that have a more modest number of mutations. And these have correspondingly a more modest response rate to checkpoint blockade, that which takes the brakes off the immune system. We were funded, my colleague Ezra Cohn and I, to study head and neck cancer in this context of personalized neoantigen-specific immunotherapy, and we first convinced ourselves by looking in the Cancer Genome Atlas that indeed the number of missense mutations, non-synonymous coding variants, is about 100 for head and neck cancer. There is, of course, a spectrum. You would have your smokers and drinkers up here and those with HPV-16 on the lower end of the mutational burden, but it gave rise to the idea that since there is a lower number of candidate antigens to test, can't we just test them all? This distinguishes this approach from that used in melanoma and lung cancer, in which the number of mutations is simply too large to test all of the candidates, and people working in that field have used epitope prediction algorithms to limit the number of mutations to something that you can actually handle and test without a complete exsanguination of your patient. So the process that we are going forward with is to have head and neck cancer patients that are enrolled at the Moores Cancer Center, and they are diagnosed with a recurrent resectable form of the disease, and these are enrolled in the immunomodulatory trials, PD-L1, and then, among others, 4-1BB. What we do then is, in an office or a clinic visit, we can get we can get a sufficient amount of nucleic acid to do our sequencing from a simple fine needle aspirate, although we're very happy to have a core or an excision when that's available. And then we get about a mil of PBL to produce the reference genome. So from these tissues, we isolate genomic DNA and total RNA, and we do whole exome sequencing on the DNA, and then we plug this into our bioinformatics section. They'll do the reassembly and the quality control and call all of the variants. We'll confirm then that mutations of which we have interest are actually expressed in that tumor by doing RNA-seq. And then comes the immunology, where we synthesize the peptides that contain the mutations and test them for reactivity with the patient's own T cells using ELISpot assay. And then when the number of mutations is large, we will use the algorithm. And I'm not going to talk about this, but it's going really well. This works. I'm very happy about this. And this should give us the antigenic immunome, that set of peptides containing mutations that distinguish the tumor from the normal cell from which it arose. since the patients we're enrolling are going to get a salvage surgery, we know that we'll be able to get the tumor-infiltrating lymphocytes from this, and we can stimulate these with the tumor-specific mutations and hopefully make a better enhanced, at least with specificity, TIL product that we can then test. And it's that testing part that I actually want to talk to you guys today about, because what we wanted was a patient-derived xenograft model that which we could do the adoptive cellular therapy with our better TIL. And with human head and neck squamous cell carcinoma, it doesn't grow in vitro very well, and this is an unfortunate feature shared by a lot of tumor types, but it doesn't actually do so well in serial passage in immunocompetent or immunodeficient mice. In fact, in the hands of someone who I would consider to be clearly an expert, it's less than 20% of these samples that can be serially passaged. And so we went looking for other ways to accomplish this, and then we came to stem cells. And this is through the work of Richard Schlegel and Shuifeng Liu, who had published a very important paper in 2013 in this journal, in which they reported that culturing human adult epithelial cells, terminally differentiated cells, in a Rho-associated kinase inhibitor, and then irradiated 323J2 fibroblasts, which have been used as a feeder cell to support stem cells for a number of studies, that these had the effect of de-differentiating, if I can use that term with you, these cells into something that looks like an adult stem cell. And you could then bypass the Hayflick limit, and that is, as you all know, cells have a certain number of divisions that they can accomplish if they've not been transformed, and most cells in culture will undergo senescence. And there are a number of cell types that can exceed the Hayflick limit and not be limited by this, and these conditionally reprogrammed cells can achieve that also. What Drs. Liu and Schlegel have shown is that you need both the feeders and the ROCK inhibitor, and that without one or the other, you don't have this essentially unlimited doubling potential. Their work has shown that the cells don't become tumors, they don't express the canonical Yamamoto stem cell factors, their karyotypes are normal, and the whole process is conditional. Withdrawing these cells from the ROCK inhibitor and the feeder cells allows them to return to the fully differentiated state that they started at. So it's something akin to a miracle for me, and the mechanism is not really understood, although it's known that the Rho kinase inhibitor that we use prevents cells from undergoing disattachment-induced apoptosis, a wonderful Greek word, anoikos, that means without a home. The feeder cells are thought to induce telomerase and MYC is important in the cell fate decision from a true stem cell to a transit-amplifying cell. And the ROCK inhibitor has a lot of effects, obviously, on the cytoskeleton, but also in the P16 retinoblastoma pathway. What I was saying about the TA cells is that if you think about epithelial cell biology, there's a basal cell that gives rise to all of the suprabasal layers that differentiate as they rise, and then the transit-amplifying cells and the stem cells coexist in that basal layer. Okay, so we put this to work in our system, and we are able to obtain from the surgeons in the cancer center fresh, living, not cryopreserved primary tumor tissue. And here's one of our patients for the medically oriented people in the audience. And we could put them into co-culture with the 3T3 cells and the squamous cells, and you can clearly differentiate both of these types. And this makes the head and neck cancer-derived cells grow, and they really grow. And it's quite surprising how quickly we can generate wonderful colonies. And then we've been able to show that following the work of Schlegel, we don't have to co-culture human and mouse cells. We can actually use conditioned media from the 3T3J2 fibroblasts, and that'll also support the growth of these cells. And after a few weeks, they start to grow as protospheroids. They're no longer attached to the culture flask. And so this is something that we've been able to do fairly regularly without any trouble on a great number of patients. So I'm going to introduce another patient that that has a couple of masses from which we isolated primary fresh tumor. And what you're looking at here is the class 1 low but positive tumors and then the unstained control there. The freshly isolated population tumor has a small population, 10 to 15%, of cells that express a very high level of CD44, and these co-express, at least in half of them, very high levels of EGFR and EpCAM, which is a lineage marker for these cells. And then we are obviously looking for stem cell markers. CD44 at high levels and high levels of EGFR have been reported to find stem cells in head and neck squamous carcinoma, and then looking at the unstained controls here. And so it would be attractive to imagine that this is a small population of cancer stem cells, but it's a tough thing to do if you don't have a model in which you can actually grow and establish those. These are the conditionally reprogrammed cells that we get from a couple of patients, and they also have low levels but present levels of class 1 MHC, but almost as a population, a much higher population of CD44 high cells that have very high levels of EGFR here and high levels there. They express EpCAM, and they also don't express CD133, and then the 323 cells are there. If you put these into an NSG mouse, they actually grow tumors, which was very nice for us to see. And then when you look at what grows out, this sort of recapitulates what we're seeing from the freshly isolated human tumor, in that there is a subset of CD44 high cells, and these co-express very high levels of EGFR, and about half of them are positive for EpCAM, no CD133, and then these are all expressing high levels, or they're expressing human class 1. In the CD44 lower population, you're seeing a bit of human cells, but mostly mouse cells. And you would imagine that a tumor, even an NSG mouse, is made up of transformed cells and a lot of stromal cells and fibroblasts and whatnot. This is not a humanized NSG mouse, so there is no human immune system component except that might have come along with the tumor, but this is these are all gone in this setting. So we wondered whether the CD44 positive cells are dedifferentiating to give these CD44 low cells, and this is what you would expect in a cancer stem cell phenomenon. So we put the CRCs into culture with normal medium and watched to see if they would dedifferentiate. This is in constant ROCK inhibitor and conditioned media, very high levels of CD44, and high levels of EGFR and EpCAM. And interestingly, they do express PD-L1, which is one of these immune checkpoint molecules. This will actually paralyze a T cell that is PD-1 positive if it interacts with this cell. Once you shift them to normal media, you can already see that the mean fluorescence intensity is going down as a population for these cells with respect to CD4, and you see the eventual formation of this second population of cells that is reduced for EGFR, EpCAM, and PD-1. A real surprise for us was what happens when you mix a 3-month-old TIL culture. So these are tumor-infiltrating lymphocytes maintained on IL-2 and nothing but IL-2 for 3 months. And then when co-cultured with that patient's own CRC, 10% of the CD8 cells are making TNF and interferon gamma. And this is surprising because these TIL actually contain T cells specific for antigens that are continued to be expressed on these cancer stem cells. We've taken this model one step further then to give us an in vivo model of adoptive cell therapy, and we've taken the CRCs and transduced them with a lentivirus that contains luciferase and GFP, and then we sorted on the GFP positive cells here, and these also form tumors in mice, and they produce luciferase signals, so we can monitor tumor burden with a CCD device. And happily, they express the same markers, EGFR, EpCAM, and CD133, in the transduced cells here as they do in the parental cells here. So at this point, this is obviously a work in progress, but we have the ability to generate, with great efficiency, cell lines from each one of our patients from their primary head and neck cancer. We seem to have cancer stem cell markers and immune escape markers, and they appear to mimic what we're seeing in the freshly isolated tumor in what comes out of the NSG mouse. And very interestingly for me as an immunologist, these CRCs are recognized by autologous TIL. Where we're going with this and currently is to look at the degree of overlap between the genes expressed and especially those containing mutations with the fresh tumor and the CRC. And although I haven't got the data in the talk, there is an encouraging degree of overlap there. We have to, of course, formally show that these cancer stem cells or that these CRCs do function as cancer stem cells, and that'll involve separating them by markers from a fresh tumor and showing which one has a greater or lesser degree to initiate tumor formation. And then we are now working on performing and optimizing adoptive immunotherapy with neoantigen-enhanced TIL against a patient's own cell line in NSG mouse, and kind of an avatar of what we'll be doing in patients. And then since we have the interaction between the CRC and the TIL, we can identify the antigens, also drawing on our neoantigen sequencing work that's already in progress. And then most excitingly, have a platform from which to clone TCRs that are from TIL, recognizing neoantigens early, very early in the TIL process before the repertoire is changed by in vitro culture. So I might have said me a lot, but the people that actually did this work have to be thanked. And all of the data that I've shown you was done by an especially talented clinical oncologist who's doing a postdoc in my lab, Aaron Miller, ably assisted by Milad Baumenhoff, who's going to follow the same path to an MD Ph.D. as Aaron. And these guys have really knocked this out in a surprisingly short period of time, and it's very interesting and brand new biology. I get a lot of help and samples from all of these nice people at the Moores Cancer Center, bioinformatics and sequencing work from our colleagues there, and then sequence done at IGM. And actually, that's the biorepository. That's my one error on this. And funding from the Immunotherapy Foundation and an NIH R01. Thank you for your time and attention.
In terms of the presentation at this session, it was very interesting, but no one's really looking at the epigenetics of modeling of a healthy phenotype that's able to actually elude mutagenesis rather than looking at strategies after this has occurred. Is anyone in your groups looking at what are the actual epigenetics of a phenotype that's able to robustly mitigate mutagenesis before it becomes problematic, rather than looking at some of the pathogenicity that's associated with it?
Well, I'm on. The answer is no. We're only people only come on our radar screen when they presented to an oncologist with cancer, and the goal of this project is to identify the patient-specific neoantigens that can be used in a vaccine or an immunotherapy. As far as a pre-existing propensity to mutation or the inability to detect that, we're not doing that kind of work.
Well, in terms of vaccine, we all know that there's so much heterogeneity in a particular mutagenic phenomena that a vaccine will never occur unless a vaccine has a much more robust level of understanding than we currently understand about this story.
Well, it's true that there's a great degree of heterogeneity in tumors, in highly mutated tumors, that's been shown. That doesn't seem to be the case from the work that we've got going in a tumor with fewer mutations. If there's fewer mutations, we're finding there's a corresponding degree of similarity between different biopsies from the same tumor that we've taken. It's early days, but that seems to be something that you can actually exploit and leverage in cancers other than melanoma, lung, and the more highly mutated ones.
Steve,
You're on.
have you been able to show that the stem cell-like cells actually have less ROC activity, or is that an epiphenomenon to make more stem cells?
I believe we're showing what can happen. I don't know from the fresh tumor which subset is actually being preserved and transformed into what we read out as CRCs. It might be a selection, or it might be that we're taking CD44 low cells and making them into CD4 high cells. I think we need to do that sort of a lineage analysis, because even sorting one subset and the other and applying this mixture is not going to give us any more than a descriptive answer.
And is anyone looking at those phenotypes as a prognostic factor in head and neck cancer?
Well, yeah, people have been doing that for a long time, but the best you can do is phenotype what is ALDH high and CD44 high and then correlate that. But to actually have a model where you can exploit that and study that, I think we're breaking into some new ground there. Thank you very much.